tend to be inherited together. The laws of probability govern Mendelian inheritance. Mendel's laws of segregation and independent assortment reflect the rules of probability. So we can't say exactly, but we can say what the probability is that you're going to get that. When tossing a coin, the outcome of one toss has no impact on the outcome of the next toss. In the same way, the alleles of one gene segregate into gametes or sex cells independently of another gene's alleles. They don't have an outcome on one another. The multiplication and addition rules apply to monohybrid crosses. So the multiplication rule states that the probability that two or more independent events will occur together is the product of their individual probabilities. So these things occur independently, but there is always a probability that they will occur together, right? So you can do the multiplication rule to determine that. So the probability in an F1 monohybrid cross can be determined using the multiplication rule. Segregation is a heterozygous plant. It's like flipping a coin. Each sex cell has a half chance of carrying the dominant and a half chance of carrying the recessive. So, the example here with flipping a coin, you have a half chance of dominant, half chance of recessive. Half chance of dominant, half chance of recessive for each gamete, right? So you have a quarter chance of each of these choices: homozygous dominant, heterozygous, heterozygous, and homozygous recessive. So, the addition rule states that the probability of any one of two or more exclusive events will occur is calculated by adding their individual probabilities. This can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. So, multiplication rule determines the F1 generation in the monohybrid cross, and the addition rule determines the F2 probability in a monohybrid cross. Solving complex genetics problems with the rules of probability. We can apply the multiplication and addition rules to predict the outcome of crosses involving multiple characters, meaning you're looking at more than one character, right? You're both round and yellow, or green and bumpy. A dihybrid or other multi-character cross is equivalent to two or more independent monohybrid crosses occurring at the same time. So in calculating the chances for various genotypes, each character is considered separately. Excuse me. And then the individual probabilities are multiplied. So, probability, homozygous dominant versus heterozygous here, 1/16th chance and a 1/8th chance. And if you do these dihybrid crosses, you'll see this. These are all your possibilities, right? So the chance of at least two recessive traits is 3/8. Inheritance patterns are often more complex than predicted by simple Mendelian genetics. The relationship between genotype and phenotype is rarely as simple as in the pea plant characteristics that Mendel studied. Many heritable characters are not determined by only one gene with two alleles. However, the basic principles of segregation and independent assortment apply even to more complex patterns of inheritance. And remember, principles of segregation, meaning that the sex cells get only one of them, and then independent assortment is that the genes are not transmitted to the offspring as a package, rather they assort independently. Now, extending Mendelian genetics for a single gene. Inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations. So these situations are the exceptions. When alleles are not completely dominant or recessive, when a gene has more than two alleles, and when a gene produces multiple phenotypes. This is where you can get into eye color, because a gene can produce many physical characteristics that look different. The degrees of dominance here, right, if you're not completely dominant or recessive, you have the choices of complete dominance, is when the phenotypes of the heterozygote and dominant homozygote are identical. In incomplete dominance, the phenotype of the F1 hybrids is somewhere between the phenotypes of the two parental varieties. And then in codominance, two dominant alleles affect the phenotype in a separate, distinguishable way. So, we have red flower and a white flower on a pea plant, okay? They segregate to the sex cells differently. In the F1 generation, we have a pink flower which would be somewhere in between the phenotypes of the two parental varieties, right? Phenotype being what they look like physically, red, white, in between is going to be pink, okay? And then you can cross the F1 generation in order to get the F2 generation, and you see several. You see the red flower, then you see the incomplete dominance, and then you see the recessive, because it affects in different ways of the phenotype. So the relationship between dominance and phenotype. A dominant allele does not subdue a recessive allele. Alleles don't interact that way. Alleles are simply variations or differences in a gene's nucleotide sequence. So for any character, dominance, recessiveness, relationship of alleles depend on the level at which we examine the phenotype. Tay-Sachs disease. This is a fatal dysfunctional enzyme that causes an accumulation of lipids in the brain. And lipids, remember, are fat cells. And at the organismal level, the allele is recessive. At a biochemical level, the phenotype, example, the way the enzymes act, it's incompletely dominant. And then at the molecular level, the alleles are codominant. So depending on how you look at the phenotypes, the levels, it depends on how you are going to determine if it's dominant or recessive. Now, the frequency of dominant alleles. Dominant alleles are not necessarily more common in populations than recessive alleles, and that's a common misconception. Most people think that if an allele is dominant, then that means it occurs more often. That's not necessarily the case. For example, one baby out of 400 in the United States is born with extra fingers or toes. You would think that the gene for extra fingers and toes would be recessive. However, 1 out of 400, that's a pretty high ratio. So it doesn't mean that it's more common, the dominant is more common. The allele for this unusual trait is dominant to the allele for the more common trait of 5 digits per appendage. In this example, the recessive allele is far more prevalent in the population's dominant allele. So, the trait for being born with extra fingers and toes is dominant, and yet it occurs less, right? The recessive is the one that occurs more often, that you have individuals with 5 fingers and toes. Now, with situations with multiple alleles, most genes exist in populations in more than 2 allelic forms, which makes, for situations that are a little more complicated than Mendel's studies with the pea plants. For example, the 4 phenotypes of the ABO blood group in humans are determined by 3 alleles for the enzyme that attaches A or B carbohydrates to the red blood cells. The enzyme encoded by the IA allele adds the A carbohydrate, whereas the enzyme encoded by the IB allele adds the B carbohydrate. The enzyme encoded by the lowercase I allele, the recessive, doesn't add either one. So, the 3 alleles for the ABO blood groups and their carbohydrates. The IA adds the A carbohydrate, the IB, the B carbohydrate, and the recessive I, none. Now, blood group genotypes and phenotypes, so what they look like and what their genes are. You have several different choices, right? And you have blood cells with these A antigens on the outside, B antigens on the outside, both A, B antigens, and then no antigens if you are type O blood, okay? And this is the options that you have for your own blood type. And then you have a whole nother factor that codes for if you're positive or negative. Pleiotropy. So most genes have multiple phenotypic traits as a property called pleiotropy. For example, pleiotropic alleles are responsible for the multiple symptoms of certain hereditary diseases such as cystic fibrosis and sickle cell disease. So with something like cystic fibrosis or sickle cell disease, you don't just have one symptom. You have several different symptoms. They affect the body in different ways, and that is because of pleiotropy, meaning that the genes have many phenotypic effects. Extended Mendelian genetics for 2 or more genes. Some traits may be determined by 2 or more genes. In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. For example, in a Labrador retriever and many other mammals, coat color depends on 2 genes, not just one. One gene determines the pigment color, so we use capital B for black and lowercase b for brown, and the other gene alleles capital C and lowercase c, color and no color, determine whether the pigment will be deposited in the hair. So if you have a brown lab or a black lab, this is going to be determined by both of these genes, right? They may have the gene or the genotype for the pigment color, but if they don't have this genotype for that the color is the pigment is deposited, then it won't show up, even though they have the genes for it. Here we go. So they've done a test cross with these individuals, and here are all our options, right? And you've got this 9:3 to 4 ratio, and you can go into specifics. You shouldn't have any problem doing this doing this test cross with these different genotypes, and then you can see the phenotype is for black, brown, or blonde. Excuse me. All right, polygenic inheritance. Quantitative characters are those that vary in population along a continuum. Quantitative variation usually indicates polygenic inheritance, an additive effect of 2 or more genes on a single phenotype. Skin color in humans is an example of polygenic inheritance. So, you have a degree of skin color, right? And that is because you have 2 or more genes that determine this physical appearance, okay? And once again, rather than using letters here, they've just used colored dots, and you should be able to perform this test cross as well. So we're going to stop there for today, and we'll talk about nature and nurture, the environmental impact on phenotype tomorrow. Have a great weekend.